And this is my colleague, and we're from Pennsylvania State University, and we're going to be talking about models of versus models for heuristic to support teachers' learning of modeling practices in molecular biology research. So in the United States, we have the next generation science standards. These standards have three parts: so the disciplinary core ideas, cross-cutting concepts, and science and engineering practices, all sort of tied together with authentic phenomena or problems at the center. We are from a center at Penn State University called the Center for Science and the Schools. We focus on these practices as part of our professional development programs we create for in-service teachers, in-service science teachers primarily. And we get to leverage the science and engineering work going on at the university. We partner with science and engineering faculty, learning what it is they're doing in their research. And we build that into programs for teachers to come and learn about those practices and take back to their classroom. So we do know that science teachers don't really have a strong understanding of these practices, and they certainly don't about science, but they know even less about engineering. So we really want to learn what it is the scientists and engineers are doing as far as the actual practices and approaches that they're using in their research. And the teachers, don't have any kind of authentic experiences using these practices. If they took undergraduate science courses, as we heard from our opening speaker this morning, a lot of times they are doing verification labs where there's a known right answer at the end. And their pre-service programs don't require them to have any sort of research experience in the science field of any kind. So they're not at all familiar with these practices and don't then know how to integrate them into their classroom. So we're going to talk today about a particular program, specifically on molecular modeling in molecular biology called Shape Matters. And this is for high school teachers in both biology, and we have a few in chemistry who taught biochemistry at the high school level. So this program was funded by our National Institutes of Health. Under the National Institute of Medical Sciences, they have a program called the Science Education Partnership Award. So this was a 5-year program, and we had a multidisciplinary team put together as part of this project to build the program. So faculty from the College of Education, that's ourselves, from the Everly College of Science, and also our College of Medicine. And this was really designed to help teachers build their knowledge of science practices, but we focused in particular on developing and using models and the way that they do this in molecular biology research. So we know that this particular practice is prevalent in molecular biology research that's going on at our university. And the way teachers tend to view developing and using models is a one particular way, even though we find it's used in a myriad of ways in actual science research. They view it mostly as a way for them to use pedagogically to teach students about a particular concept or phenomenon as an illustration or a representation, rather than seeing it as a tool for the students to use as part of their inquiry or to conduct investigations in any way. These are ways that students could be using models to pose their own questions, generate data, make predictions, and communicate ideas. And we find that those are not often seen in the high school classroom. So a little bit about the modeling of versus modeling for heuristic. So what I just described, sort of teachers' views of models tend to follow these two portions. The modeling of has to do with representing something, say it's a molecule and you have the ball and stick models to represent where the atoms would reside in a particular molecule. It's to identify or illustrate you know, how this molecule might appear, even though we can't see it with the naked eye. So it's about having this one-to-one representation of particular either phenomenon or something that's being studied. The modeling for is more about using models as a tool. These are dynamic models. They can be used, like I said, to do inquiry or investigations, and in much more sophisticated ways than simply having a static representation of something. So in the Shape Matters program, we had a 2-week summer workshop. Our summertime is when teachers are out of the classroom. It's not quite a 3-month period of time that they're not teaching, and they're available to participate in professional development. So we wanted to engage them in modeling of and modeling for types of activities using the context of diabetes and sort of studying diabetes as part of the workshop. So we intentionally designed the workshop to alternate between modeling of and modeling for, so that the teachers gained experience in sort of understanding the difference between the two and thinking about how to bring modeling for into the classroom in ways that are appropriate for the students that they're teaching.
So this slide is basically just walking through how we walked back and forth between modeling of and modeling for the workshop. I'm going to go more into depth in the next few slides, but the idea here is we use modeling of in the classroom to help build teachers' content knowledge, and specifically we're focusing on diabetes, and the molecule in question was insulin. So we were using their modeling kits. A lot of the times they're like foam pieces that help to depict molecular processes from a company called 3D Molecular Designs to help build their content knowledge, in addition to using some molecular visualization software online where they did some guided tutorials to help build their knowledge and confidence of modeling in molecular biology. And then we did modeling for by giving teachers a window basically into the practice of modeling molecular biology. They work to understand how protein models are determined and built, and then to understand the data behind them and how they can use that with their students in their classroom. So to start the workshop, we started by talking about water. So for those of you who are unfamiliar with proteins and protein foldings, proteins fold in water. They fold in the cell, which is mostly water. And so the chemistry of water dictates how proteins fold. So we start with water, and the kit on the left is the water kit. And they're basically little water molecules that have magnets in them to kind of show the polarity. And then from there, we switch to modeling for by learning about how proteins are crystallized and then how they use those crystals to determine their structure. And then we went back to modeling of by using 2 different kits, the amino acid kind of mods, which are in the center. So they built amino acid, which is something they teach in their classrooms. This is something that comes directly out of kind of your freshman biology. And then how they connect together to form a polypeptide protein. And then we switched them to viewing just the functional group of the amino acids. So that's what's different in each of the 20 different amino acids using the amino acid starter kit, and how the chemistry of these functional groups determine how proteins fold. And so they then folded kind of a sample sequence. And then we went back to the crystals, and we went to the X-ray crystallography lab on campus, and they learned how X-ray crystallography is used and how the data that comes out of the X-ray crystallography is used to build a model, and what that means. And all of those models are stored on a database that students can easily access. It's called a protein database. So it's all the structures that have been determined that you can then pull up and visualize using a variety of different programs. Of course, in our program, we used one that was accessible to K-12 that had a smaller learning curve. It's called Jude. And so then we switched back to modeling up, and we did another kit from 3D Molecular Designs. It's called Insulin mRNA to Protein Kit. Basically, in this kit, they look at an mRNA gene map for insulin to kind of learn how insulin is made in the cell and how it folds. And then we switched back to the modeling for by looking at some structures related to insulin in the protein data bank and looking at, what do these structures tell us? What kind of data do I need to look at to understand where are there uncertainty in these models? How do I identify that with my students? And then doing that again with some tutorials that are guided tutorials that give them experience using some molecular visualization software. And then in the last few days of the workshop, they do an open investigation. So we use designer insulin. So things that we have, but scientists have designed to treat diabetes. So things that you might be familiar with, the one on the right poster is Humalog, which is a common diabetes insulin injectable. And they did an open investigation into the structure and function and presented what they found on one of the afternoons of the workshop. And then they transitioned into going into a research lab at Penn State with a mentor. And the idea was they were learning how research was being done in that lab to identify and tell the molecular story of that lab. And so the poster on the left is one of the molecular stories. It's on viral switches. That one of our groups of teachers did work for years. So over the course of the workshop, so the program was 5 years long, as Kathy said earlier, we had 3 different cohorts and a total of 24 participants. Most teachers who attended our workshop taught high school. We had, I believe, 1 teacher who taught at the collegiate level, but the rest were all high school teachers. 88% had a master's degree. And the majority of them, 63%, taught in Pennsylvania schools. Most of our teachers taught biology, but we did have a few chemistry teachers who attended.
So for evaluating this program, we had an external evaluator that we worked with. That particular group collected all of the data, did all of the data analysis, but we helped inform that evaluation because there were certain things that we wanted to learn about from the teachers that had participated. So this was a mixed methods, formative and summative evaluation. The group that we worked with collected teacher surveys, did project team interviews, so with ourselves, the faculty and the College of Science and Medicine, as well as teacher interviews and focus groups. They reviewed documents and other data that they collected from the teachers. And then they did both the qualitative and quantitative analysis that helped us report our findings.
So they looked at a number of different things, but one of the things they looked at was teachers' self-rated knowledge before and after the workshop, and we were particularly interested in their knowledge of the scientific practices, especially modeling. So key concepts associated with molecular biology, so their content knowledge development, and then their knowledge of current cutting-edge research projects. And again, these are all self-reported, but all of these were significant from the pre-post test. And then we also looked at their self-rated skills before and after. So their ability to be able to tell a molecular story or work on a molecular story in the classroom with their students, their ability to design and build models in Jude, which is a molecular visualization software, and then their ability to be able to make models in the classroom using a 3D printer. And again, the paired sample t-tests were statistically significant for all 3 after the workshop. And these findings were also reported, were also supported by teacher interviews and project team interviews. So, but teachers reported feeling more confident in the classroom and were able to use the skills in the classroom, were using the things that they learned in the classroom. And then project team members noted how they watched teachers grow in their knowledge from the beginning of the program to the end of the program, and also saw the results of students presenting their work in the modeling stories an event that we hosted called the Smart Team Symposium. And then we then gave at the beginning of the workshop, we gave a pre-test and a post-test that tested their knowledge on modeling, and we used a modified version of the Students Understanding Modeling Survey. And so that survey looks at 5 different domains in modeling. So models as multiple representations, models as exact replicas, the use of scientific models, the nature of models, and models as explanatory tools. And in all 5 domains, we saw growth, but the only one that was statistically significant was models as multiple representations. And then we'd also like to make a note about the models as exact replicas. So after the first year, evaluation data showed that teachers still view models as exact replicas, which is not the case. And so we made some changes to our professional development to help teachers better understand that models are not exact replicas. And our second and third cohort had bigger gains in this area, and evaluation data supported basically that the changes we made help teachers to better understand this aspect of modeling. And so some key points here is that the professional development focused on giving teachers a window into the practice of modeling, and we did that by leveraging our multidisciplinary team. So our science researchers were all molecular biologists who were very knowledgeable about the ways that work is conducted in this field, and particularly in the ways that modeling is used in this field. So they helped us to provide and create experiences for teachers that gave them a window into how modeling is done in molecular biology, while the science education experts all had classroom teaching experience and also knew best practices and ways to engage students and connect it to the current standards in Pennsylvania. And that alternating back and forth between modeling of and modeling for help teachers to better gain a better understanding of modeling practice, which is evident by the evaluation showing statistically significant growth in their understanding of the modeling practice. But we would like to point out that the findings are limited to a small sample size of teachers, and that all of our knowledge findings are self-reported. In the future, evaluation could look at specifically into classroom observation, which we did not do, to confirm what is going on in the classroom. And then we just like to take a moment to thank the NIH and faculty.
Okay.